Chapter 11, Survey of Bacteria. The objectives for this topic are: provide a rationale for the name deeply branching bacteria, explain the function of heterocysts in terms of both photosynthesis and nitrogen fixation, discuss the lack of cell walls and mycoplasmas, identify significant or detrimental effects of the genera Clostridium, Bacillus, Listeria, Lactobacillus, Streptococcus, and Staphylococcus. Explain the slow growth of Mycobacterium. Identify significant beneficial or detrimental properties of the genera Corynebacterium, Mycobacterium, Actinomyces, Nocardia, and Streptomyces. Describe the appearance and function of prosthetic and alpha proteobacteria. Name 3 pathogenic and 3 useful beta protobacteria. Describe the gamma proteobacteria. List several members of delta proteobacteria. Describe the unique features of chlamydia and spirochetes, and describe the ecological importance of bacteroides. The subtopics for this topic are deeply branching bacteria, phototrophic bacteria, low G plus C gram-positive bacteria, high G plus C gram-positive bacteria, gram-negative proteobacteria, and other gram-negative bacteria. First, let's discuss the deeply branching bacteria. And to do this, let's discuss the relatedness of various organisms on Earth and how we would diagram the relatedness of these organisms by using a phylogenetic tree. So first off, there was an ancestral cell, or a population of ancestral cells. The reason why we have this hypothesis is because all organisms use the same DNA code. You can take DNA from one type of cell, say a bacterium, and put it into another type of cell, say a eukaryotic plant cell. And the plant cell will make the bacterial protein. And we'll be talking more about that later on in this topic. But the shared or universal codon code indicates that all cells are descended from one ancestral cell. Eventually, this ancestral cell split into 2 populations: the bacteria and the ancestors of the archaea and the eukaryotes. Now, eventually, the archaea and the eukaryotes split into 2 different groups. They became different enough that they have their own domains. Now, bacteria did not remain static. They also changed over time. And we have most bacteria over here, but over here we have bacteria that live in environments that are very like what the early Earth would be. Non-oxygen, very hot, acidic, high radiation, and so because they kind of took off on a side branch from most of the bacteria, we call them deeply branching bacteria. That's where the term comes from. The example that I want to use of deeply branching bacteria is Deinococcus radiodurans. Was originally isolated from nuclear power plants. These organisms have a tendency to form tetrads, and they are very resistant to radiation, as evidenced by the fact of where we originally isolated them. They have wonderful DNA repair enzymes. And of course, we're interested in these DNA repair enzymes because we'd like to know how to do the same thing. If we could protect people from radiation, say ultraviolet radiation, so that you don't get skin cancer, that would be a wonderful thing. Next, we're going to cover phototrophic bacteria, or bacteria that photosynthesize. We're going to review photosynthesis, at least the light-dependent reactions, and then we're going to talk about cyanobacteria in some detail. You may recognize this diagram from when we discussed anabolic reactions, but let's go over it. So in non-cyclic photophosphorylation, light comes in and excites an electron in the magnesium of chlorophyll A. And the electron goes through an electron transport chain, eventually ends up in reaction center 2. hydrogens are pumped from one side of the thylakoid membrane to the other side. Then this electron gets passed on. Eventually the terminal electron acceptor is NADP+, goes on to become NADPH, which goes on to the Calvin-Benson cycle. Also, all of these hydrogens that are pumped to the other side of the membrane go through ATP synthase, which combines ADP and inorganic phosphate to form ATP, which also goes to the Calvin-Benson cycle. All of these energy-carrying molecules aid in fixation of CO2 into organic carbon chains. Now, this electron over here in photosystem 1 needs to be replaced, otherwise we can't continue the electron transport chain. So water is split. The electron from water goes here to replace the electron that has been removed from the magnesium in chlorophyll A, and we end up with hydrogen ions and oxygen. Now we'll talk more about this when we talk about the cyanobacteria, but this process of non-cyclic photophosphorylation results in the release of oxygen, which now is a good thing. But if you take a look at the geology of early Earth rocks, the atmosphere did not have a lot of free oxygen. We had what was called a reducing atmosphere. We still have phototrophic bacteria that do not release oxygen. They are the green non-sulfur bacteria, the green sulfur bacteria, purple non-sulfur bacteria, and purple sulfur bacteria. Now I'm going to talk about the green and purple sulfur bacteria because it dovetails so nicely with the oxygen releasing system. They have a very similar electron transport chain to what oxygen releasing phototrophs have, and so they still need to replace the electron that's removed from their equivalent of photosystem 2. So the question is, where do they get that electron? Well, they use hydrogen sulfide instead of water. See how it's very similar to water? 2 hydrogens and a sulfur instead of an oxygen? Well, their equivalent of photosystem 2 splits the hydrogen sulfide. We get hydrogen ions, just like in oxygen-releasing phototrophs, but instead of producing oxygen, they produce elemental sulfur. Purple sulfur bacteria keep the sulfur in granules inside of them. Green sulfur bacteria export the sulfur, and so they have sulfur deposits around them. Interesting, huh? And, of course, all non-oxygen releasing phototrophic bacteria are obligate anaerobes. Next, let's move on to the cyanobacteria. They undergo oxygenic photosynthesis or photosynthesis that releases oxygen. Now, from geological records, the cyanobacteria are credited with changing our atmosphere from a reducing atmosphere to a oxygen-containing atmosphere, of which we are breathing today. And they used to be called blue-green algae. You can see that they're a little more blue than the usual green of, say, algae in plants, but they're not algae. They are prokaryotes. They do not have a nucleus, so their name was changed to cyanobacteria. And later on, when we get to the next chapter, when we talk about the different types of algae, we will be talking about cyanobacteria more because they are very closely related to chloroplasts. The thought is that an ancestor or a cell from the same population that gave rise to cyanobacteria was engulfed by a eukaryotic cell, became adapted to the interior of that cell, and became chloroplasts. Now, cyanobacteria are important because they release oxygen. We all like getting fresh oxygen to be able to breathe. Also, they fix nitrogen. Now, remember when we talked about nitrogen fixation in previous chapters, most of our atmosphere is atmospheric nitrogen, but it's got a triple bond that is very, very hard to break, and very few organisms can break that bond and take atmospheric hydrogen and convert it into ammonia, which is easily used by quite a few organisms. Now there's a problem with the process, though, of fixing nitrogen. Nitrogen fixation does not proceed in the presence of oxygen. The enzyme in cyanobacteria that breaks that triple bond in atmospheric nitrogen is killed by oxygen. So you've got this organism that is releasing oxygen, yet to be able to fix nitrogen, to come up with the ammonia that it needs for its proteins and its nucleic acids, it can't be in the presence of oxygen. That seems like a catch-22 to me. I don't know about you guys. Well, they solved this problem, at least in this species, with a structure called heterosis. And there's one right there. It's a cell that is specialized in fixing nitrogen, and it walls itself off from atmospheric oxygen. These cells here are photosynthesizing, so the sugars that are produced by the photosynthetic cells are moved over to the heterosis. The heterosis export ammonia to the other cells. So it's a community. Now, other species of cyanobacteria solve the problem in a different way. They photosynthesize by day, and they fix nitrogen by night. Very clever, isn't it? Here is a summary of the various phototrophic bacteria, and it wraps up our discussion of photosynthetic bacteria very nicely.